got it. Share some slides. Go back to the top. All right, so here's what I'm gonna do. I'm gonna stop the share. Okay, so hello everybody. We made it through week 1, just about. So we're gonna cover chapter 4 today, and we're gonna take our quiz from 11 to 11:30. Go ahead and ignore that email that I sent out about using the Respondus lockdown browser. I thought that it was available for you all on your side. I can see it on my end, but if you can't, we're going to do that for quiz 2 next week. For today, let's just stay logged in to HD meetings or log back in to our class link, so that way I can monitor everybody through their webcam. It just, I guess I could record that for my own sake. So please come in from 11 to 11:30 if you do log out after lecture, so that we can take our quiz with a webcam so I can monitor you. And let's see, about the quiz. So I said it was going to be 12 questions. When I wrote it yesterday, I wrote 12, and then I took off the 2 hardest questions. So now it's only going to be 10 questions, and your quizzes are going to be more straightforward. Just, did you retain what we talked about in class? The exams are going to be a little bit more difficult, a little bit more critical thinking required for those questions. But again, today it's only 10 questions. You'll get 30 minutes. It's more than enough time. And then keep in mind, each quiz is worth 5% of your final grade. So every question today is worth half of a final point for your grade. So today's lecture will probably take maybe 45 minutes to an hour. I do want to start and see if anybody has questions before we get rolling, though. So y'all in here, do you have anything?
Who's the other guy with Charles Darwin that you mentioned yesterday?
Alfred Russel Wallace.
I can't get my camera to work.
You can't get your camera to work. Okay.
Yeah.
Let's see. Do you have a phone? With
I do,
HD
yeah. I do have a phone.
Yeah. So, yeah, you could just stream through your telephone.
Okay, I'll do that.
I do. I'm sorry, Professor Nick.
Yeah.
This is Latoya. I do have a question about that, taking the test on my phone. My computer is doing some weird things, and I don't know exactly how I'm going to take the test on my phone.
Okay. So it is multiple choice.
Okay.
So if you go into Canvas just through your web browser, you should work just fine. You don't need an app to take it, so long as you have internet access.
Okay.
I would prefer that you had HD meetings so that you can stream your video footage.
Yeah, that's what I'm on now.
Okay, yeah, great. I can see you, you're fine. So while you're taking your quiz, you should be able to just open up your web browser, and then I should be able to basically just see that you're not using your textbook while you take your exam or your quiz.
Okay.
And since you'll be logged into HD meetings, if you need me, you just say something and I'll be right here during the whole time.
Okay. Okay.
Yeah, worst case scenario, we'll deal with it as we go. With an online class, we're going to have to do some sort of troubleshooting throughout the semester, I guarantee it.
Oh, yeah, I'm sure.
Yeah, let's just be patient and enjoy it.
Okay.
So does anybody have questions about the quiz or chapters 1 through 4 material before we get started?
I had a question about chapter 3. I was really confused about what it was all over.
Okay, so chapter 3, let's pull this up. One sec. Share. All right, so chapter 3 was mostly about how we are going to study microorganisms. So we talked about taxonomy, so how we group organisms. We talked about the history of those groupings. So originally we had just plants and animals. Then we had a unicellular category, the protists. And then after that, we had the Monera. And then from there, we also had the fungi as their own taxonomic organization. So the organization of life is a part of this. Let's see. So we have the domain, kingdom, down to species level. So most inclusive being the domain, the most exclusive, or we could say the least inclusive, the most specific is a species. And then we talked a lot about microscopy. So at the end of this presentation, there's a table that is going to summarize the different types of microscopy and their magnifications. But what I want you to know is why we're going to use one type of microscopy over another. So for example, if you're doing something like a gram stain, you would only use a light microscope to visualize your stain technique. I want you to know that. It's very practical. You will come across it in lab. And so that was the majority of what we talked about. We talked a lot about microscopy and taxonomic organization. So did you all have any specific questions about any of that content?
Can you go over the gram test one more time?
Yeah, I'd love to.
Thank you.
Yep. Okay, so the way that a gram stain is going to work is we're going to have bacterial cells. We're going to place them on a glass slide. The glass slide is going to be all the way see-through. It's transparent. So now we can shine a light through our sample. So the bacteria go on a glass slide, and then we apply heat. That heat is going to trap the bacteria on the glass. It's also going to kill them. So the first step in a gram stain is to use crystal violet. It's a blue stain. It is a simple stain, which means that it carries a positive charge. It's a positive charge with a blue dye attached to it. Bacteria carry a negative charge. The negative interacts with a positive, so we get blue on our bacterial cells. All cells are going to be blue. The next, we're going to add iodine. Iodine, you can think of this as mortar for the crystal violet. It's going to help keep the crystal violet on the outer portion of the bacterial cell wall. The third step is called an alcohol wash. So only gram-negative bacteria will have their blue color removed. That's because their outer layer contains lipids. These lipids are soluble, or they will dissolve when they're exposed to ethanol. Gram-positive bacteria lack these outer fatty acids, these outer lipids, so the ethanol does not remove the crystal violet and the iodine that's bound to the outer layer called peptidoglycan. So they're going to keep their purple color. And then, so if you're a gram-negative cell and you've lost your purple color, the purple color carries a positive charge. So if your positive charge is gone, now you're available to receive another simple stain. So we're going to do what's called a counterstain, where we put a red dye with a positive charge on it. If your negative charge is available, then you will become red after the counterstain. Safranin does not influence the color of gram-positive bacteria because their negative charge is already saturated entirely with the positive charge blue dye. So that's why we end up with gram-positives being a blue-purple color and then gram-negatives being a red-orangish color.
Hey, Nick, how do you know which one to use?
So you're going to do this, the same technique, always, whether you know what you have or whether you don't know what you have, because let's say that you know what you have. You know you have E. coli is gram-negative. Well, if you're going to do an experiment on it, you want to know that that's all that you have. So if it's gram-negative, but you get some blue cells, you're not going to do that experiment because it'll show you that you have some sort of contamination in your sample. So you're going to do these same 4 steps regardless of which organisms you think you have or you know you have.
Gotcha.
Great. Pull that up for me. Thanks.
Thank you.
Yeah, you're welcome. Is there anything else you wanted me to cover before we get started?
No, I'm okay right now. Thank you.
All right, great. So let's go ahead and go into chapter 4. All right, like I said, this is going to take maybe 45 minutes to an hour. So we talked about 3 major domains yesterday. Again, domain being the most inclusive, the largest umbrella term for a taxonomic organization. The 3 that we talked about were Archaea, bacteria, and eukarya. We're not going to talk about eukarya today. We're going to focus mostly on bacteria because we don't know of any archaeal species that infect humans or anything else for that matter. Within bacteria, we have 25 phyla recognized, but I'm sure that there are more. We've just only discovered and organized 25 phyla. So the book mentions some terms that I just want to clear out a little bit. They mention that a certain type of pathogen is an obligate intracellular organism without going into an explanation of what those terms mean. So let me explain that. Obligate means that it is required. You are obligated to do something. You must have it. That is in opposition to facultative, which means you can. Intracellular means within a cell, and extracellular means outside of a cell. So an example from the textbook, they say that Rickettsia is an obligate intracellular parasite. That means that it can only reproduce within another cell. It can survive in the environment, but it won't reproduce unless it's in another cell. Some organisms can't survive within other cells. Others can, but don't prefer it. So we would call that, say, a facultative intracellular pathogen. All right, so now we're just going to go through some general types of bacteria. You don't need to memorize exactly which organism belongs to which group. I just want you to know one little fact, one little keyword about each type of bacteria. So the proteobacteria, these are the largest, most abundant group. These are all gram-negatives. This is where E. coli fits into. And we think that this is the likely origin for where mitochondria so this is a gram stain. They show up red or pink. All right. We're getting an unstable internet connection. Let's hope it doesn't cut out on us. The firmicutes, these are gram-positive organisms. We have bacillus, clostridium, staphylococcus, streptococcus. Again, you don't need to memorize these, but you will start to organize it all as we go throughout the semester and as you work in your lab. You'll come across these names again. The Actinobacteria, these are abundantly found in the soil. And this is where we're going to get antibiotics such as streptomycin. So there's a group called the Streptomyces as a genus, and they produce an antibiotic. So think about what soil is. Soil is full of microbial life, and they are in direct competition with one another. So if you're a soil microbe and there's another organism over here that you have to outcompete for nutrients, you can create something that can eliminate that organism so that you can take the resources that it would take. We can harness that and use it in humans. That is an antibiotic. An antibiotic is just one chemical that will kill another organism. And one thing I'm going to try and do throughout this course is help educate you all about what different antibiotics do, because not all antibiotics mechanically operate the same way. So we'll mention a few of those today. These cyanobacteria are special because they can carry out photosynthesis using chlorophyll. So again, keep in mind photosynthesis is harnessing the energy of the sun through UV rays. We're going to convert, pardon me, carbon dioxide into oxygen and produce energy for the cell. And these can have various forms, whether they're on their own as a single cell, they can be in filamentous or branching forms as well. And so cyanobacteria, in the book, they mention that they're also called blue-green algae sometimes, but that can be misleading. Let's avoid that term and let's just call them cyanobacteria. So these can be black, yellow, green, or red. The textbook has a nice image of fields that show up red due to blooming events. But again, these are special because they're carrying out photosynthesis. And if I recall correctly, I believe this is the likely origin of chloroplasts due to endosymbiosis. The Chlamydiae, these are really small. They're often pathogens in humans. These require intracellular invasion in order to replicate. One example of this is Chlamydia trachomatis. This causes chlamydia in humans. The book mentions that this is the most commonly treatable sexually transmitted disease. Spirochetes also can cause some sexually transmitted diseases. They can also be present in the mouth to cause some gum disease. So Treponema pallidum is a spirochete that causes syphilis. In the mouth, we have an organism called Treponema denticola that can play a role in causing gum disease. So the whole point there is just that we can have organisms in the urogenital region or in the mouth that are somewhat related to one another and can cause disease in humans. So these spirochetes, they look like little corkscrews that spin to produce movement. And we'll have an image of their flagella later in the lecture. So we mentioned Treponema pallidum. There's also the organism that causes Lyme disease, Borrelia burgdorferi. Long name there. So this is the bacterium that causes Lyme disease. So this is carried by ticks. And you'll have seen, or maybe you may not have, whenever you get bitten by a tick that is carrying this Borrelia organism, you get what's called a bullseye pattern from the site of infection. So keep in mind, these are corkscrew-like shaped organisms. That allows them to move really fast through our bodies. So what this bullseye pattern is, in the middle, we have the bite. So where the tick bit you and infected you with the spirochete. That spirochete is whipping around and swimming through your body. Your immune system is chasing it, but it's faster than your immune cells. So that's how we end up with this ring. Notice that it radiates out from the site of infection. The ring is your immune response chasing the bacteria as they swim away from the site of infection. This is pretty cool. Yeah. And you can literally watch videos through a microscope of these organisms swimming and outrunning your immune cells. So if you're in a hospital and you see a bullseye pattern, think Borrelia, Lyme disease, spirochete. We have a slide for the moment about Archaea. Again, we're focusing mostly on bacteria. This is a microbiology class for health science. So we have two major organizations within Archaea. The Archaea include methanogens. So these are going to live in areas with low oxygen, like the rumen of a cow's stomach. And they're going to produce methane, CH4. These contribute to global warming. So this is one reason why we need to get away from these large factory farming operations with cows. You all may have heard of CAFOs. That is a concentrated animal feeding operation. So if you have a lot of cows in one place, let's say you cut down trees in the Amazon to start raising cows, you're getting rid of the ability to photosynthesize with the trees, and you're producing more methane. So you're just ramping up global warming like crazy. That's a big problem. We also have extreme halophiles. Keep in mind, phile means to love, halo means salt. So things like the Dead Sea, areas like the Dead Sea with just immense amounts of salt, certain organisms within this group can survive there. The hyperthermophiles, so hyper means a lot, thermo, heat, philes. They love a lot of heat. So these are going to grow at high temps, specifically when there's low pH. Remind me, low pH. Is that from 0 to 7 or 7 to 14?
0 to 7.
0 to 7, is that acidic or basic? Acidic. Perfect. From 7 to 14 is basic or alkaline. Alkaline and basic mean the same thing. Okay, so one organization was the Archaea. We also have the Archaea. Try and say that 5 times. So these are going to often grow in really hot, acidic environments, again, low pH, but some of them are also found really deep in cold parts of the ocean. So just know that these can survive in all sorts of regions, and they're very adaptable. By the way, I did get an email from somebody saying that I was going really fast in lecture. So as I talk, I'm going to check in and make sure we don't have questions. But if you need me to slow down or repeat anything, please just interject, just say something. Keep in mind that I cannot see your all's video while I'm giving this lecture, okay? Okay, let's talk about some stuff that you're definitely going to see in the upcoming weeks in your lab. So we can describe prokaryotes according to their cell shape. We're going to call that shape the morphology. And then how the cells are organized in relation to one another is called the arrangement. So let's start with morphologies. So a bacillus is a rod-shaped organism. Take a look up here at part A in this figure. You can see these look like little Tic Tacs, right? So they're rod-shaped bacteria, and they can exist in 3 forms: as a single cell, as a pair of cells, or in a chain. So the way this is going to happen is the cell is going to undergo binary fission, it'll divide, and then it just won't separate if it's a diplobacillus. Di means 2, diplobacillus, 2 bacterial cells. If we have a chain, or streptobacillus, then we continue to have binary fission events along the chain without separation of the cells. And these chains will go on. I'm not sure at what point the bacteria will stop forming a chain. That probably depends on the species in the environment. So with bacilli, we have 3 possible shapes. Round bacteria we're going to call cocci. Coccus, C-O-C-C-U-S, is the singular. Cocci is plural, referring to more than one coccus cell. So these can exist in the most abundant variety of different cellular arrangements. Notice that they can be single cells, they can be pairs. We can have a group of pairs in 4 cells called a tetrad. We can have chains just like the bacilli, or we can have a cluster that looks like a bunch of grapes. We're going to call that staphylococcus. So here the staphylo portion refers to the arrangement, coccus refers to the morphology. And then at the bottom in part C, we have our spiral-shaped bacteria. These all exist only in a single cell arrangement. So the spiral shape have the least number of cellular arrangements, and the cocci are the most diverse type of arrangements. Our Vibrio, I've seen this described as, it looks kind of like a kidney bean. It's just kind of like an oblong oval. We have spirilla, and then our spirochetes are the ones that we talked about that have that nice corkscrew shape, with the spirilla being somewhat in between the Vibrio and the spirochetes. But again, these are all only single cells. We don't have chains of spirilla. Okay, the book doesn't include this 8-cell arrangement of cocci called a sarcina. I believe I'm pronouncing that correctly. It's like having 2 tetrads that are bound to one another. So you have a cube of circular cells now. Not the most important, but it's worth mentioning. Again, you might see it in lab. So this slide is to help you step back and organize all the material that we've talked about to this point. Not all of it, but a bit, right? So bacillus is a cellular morphology. It means a rod-shaped bacterium. It's also a genus of bacteria. So there's one species called, for example, Bacillus subtilis. Bacillus is the genus, subtilis is the species, correct. But it also has a morphology that is rod-shaped. So if you see it being italicized with a capital letter, we're referring to the genus. If you see it in lowercase without the capital letter, we're referring to it as a morphology. The same goes with Staphylococcus and Streptococcus. These are both genera, which is the plural of a genus, as well as cellular arrangements. So, somebody online, tell me what a Staphylococcus is going to look like. What is its shape, and then what is its arrangement?
It looks like it's in a triangle shape.
Okay, so that is in this image, but generally speaking, can you recall what staphylo refers to?
It means a cluster.
There you go, right. We're gonna have a cluster of round cells. So staphylo means cluster, coccus, round cells. In this image, it looks like it's a triangle, but just think it's not a chain and it's not organized in a tetrad. The book describes it as some sort of random orientation. I view it best as a grape-like cluster. I think that's a good analogy. And then Streptococcus is going to be a chain of round cells. Strepto chain. Okay, so within bacterial cells, they aren't as highly organized as eukaryotic cells because we don't have these subcellular compartments with membrane-bound organelles. We don't have a nucleus, but they are ordered, both on the inside and the outside. We're going to talk about some sensing components later when we talk about pili. We have compartmentalization of metabolism. That means we have a cell envelope. When we talk about an envelope, that includes the plasma membrane and the cell wall together. So we're going to separate our inside from the external environment. That's compartmentalization. And we're going to do this as we grow, as we reproduce. Keep in mind that all cells are going to use DNA and must reproduce to contribute to the next generation. Okay, so in this next slide, I'm going to blow up this image so that we can look at it a little bit closer. But let's mention pili. So a pilus is singular, pili is plural. If you come across the word fimbriae, that is synonymous with pili. So these are going to project out of a bacterial cell, but they're not used for movement like flagella. We'll talk about flagella, but pili are for sensing things in the external environment, not motility. And so these are made up of proteins, and at the end of them, they can have what's called an adhesin. An adhesin is going to do what it sounds like. It provides the ability to adhere or for adhesion. It's going to allow a bacterial cell to bind to something else, whether that be a surface or another bacterial cell, so that it can form a biofilm. They can also be used to transfer genetic material from one organism to another. This is going to be through something called a conjugation pilus. So one organism can inject DNA into another organism through a conjugation pilus. All right, so the book mentions these inclusions. So these are wrapped in proteins, and we can have certain nutrients that could be limited, phosphate and calcium. We could have some glycogen in there. Speaking of sugars, we have a glycocalyx. So this is an exterior sugary coating. It can either be really thick or a little bit thinner, and I have that on a slide upcoming. What is important for you to take from this slide is that pili are distinct from flagella. Flagella provide motility. Pili are going to allow for adhesion and genetic exchange. Keep in mind that those are structurally different, those that do genetic exchange and those that are for adhesion. But the pili can do both depending on the type. And then let's also keep in mind that we do not have a nucleus in our bacterial cells. The DNA is going to be within the cytoplasm in a region called the nucleoid. All right, so let's talk about flagella and how they're built, what they do. So these are, think of it as a tail for bacteria to move through their environment. The book mentions how small bacteria are compared to us. And they're saying that it's hard for bacteria to swim, to move through water because they're so small. So water is going to be really difficult for them to move through. They equated it to us trying to swim through molasses. But they're able to do it. So there are three parts. We have a helical filament. So this is the outer part that whips around, the long part that we visualize. There's a hook in the middle that is going to connect that filament, which is made of proteins. So the hook is going to bind the filament into the cell wall to a part called the rotating basal body. So this rotating basal body is going to span the cell wall as well as the plasma membrane. Note here, one difference is that, so in all bacteria, we're going to have a two-disc system in the cell membrane. In gram-negative bacteria, we have a two-disc system in the cell wall, whereas gram-positives are only going to have one ring in their cell wall. So that's just, a difference to take note of. Again, keep in mind that not all bacteria have flagella. So when I say gram-negatives have this, gram-positives have that, when I say that, I'm referring to some gram-negatives and some gram-positives. Other organisms are non-motile, meaning that they cannot move intentionally. So when bacteria move, we're going to refer to that as motility. If they move towards a chemical or a nutrient, we're going to call that chemotaxis. They are moving towards a chemical. Pardon me, and they're going to move up a concentration gradient. So that means that they're going to go from low concentration, they're moving towards high because they want that nutrient. Why would you swim towards something that is less available? No, you're going to move towards something that is abundant. And they can swim very fast, so these flagella can rotate up to 1,500 times a second. That allows them to move up to 10 body lengths per second. The book mentions that the fastest human can only move about 5 or 6 body lengths per second. So these can go very efficiently through even a molasses-like substance for them. Really impressive. So the way that they do this is through 2 different mechanisms. So the flagella is either going to spin counterclockwise or clockwise. If it spins counterclockwise, we're going to call that running. It makes them go forward, straight and narrow. And then, let's say that a nutrient is depleted and they need to switch up directions. Then the flagella spins clockwise, and they're going to do what's called tumbling, which means they're not going forward, they're just kind of moving chaotically until the flagella starts to spin counterclockwise again, and then they move forward.
What was counterclockwise called again?
Running. Yeah. So just think like you're running straight forward. Counterclockwise, tumbling is clockwise. All right, so we mentioned spirochetes earlier. This shows how their flagella is organized. So not all flagella are going to be exterior structures. Spirochetes have what's called a periplasmic flagella. So periplasm, meaning it is between regions of a cell wall. So this is encapsulated by the outer membrane, so it doesn't stick out of the cell at all. And so it goes from end to end, and as it twists, it causes torsion, and this is how the cells are going to corkscrew through. So like our Borrelia have this type of flagella to cause that bullseye pattern. It allows them to swim really fast. And so the term that they use here is endoflagella. So it's a flagellum that is within the cell instead of outside. We touched on the glycocalyx. If you see glyco, think glucose, think sugar. Glyco, sugar. And then I believe that calyx refers to exterior. So we have an exterior sugary substance. And sometimes it has some proteins in it, but it is mostly sugar. So we have 2 types. One is called a capsule. The capsule is really big, is very thick, as opposed to a thinner layer, which we're going to call a slime layer. So why are we going to have these? So if you have a capsule, think of this as kind of like a bubble suit, essentially. So that way, things in the environment aren't touching you. You've built like a moat, like an outer protective coating. So that can protect you from environmental toxins potentially, but what I think is evolutionarily significant for our class is this can help protect cells from the immune system. So it's a barrier against the thing that can hunt you down and kill you. And then the slime layer can be used to stick to surfaces. So again, if you're coated in sugars, just like, say, a lollipop that you've licked and left somewhere, it'll be very sticky and can help you stick to a surface like a tooth. And that's useful for the initial stages of biofilm formation, which was attachment to a surface. You can't form a biofilm unless you attach A sticky exterior glycocalyx can help you adhere. And then other organisms could come in with their pili and stick to you since you've stuck to your surface.
I'm assuming that's like cavities and stuff, like that.
So cavities are caused by acidic production in the mouth. So if we eat sugars, the sugars get converted to acids, and those acids break down our enamel. That's a cavity. It is caused by bacteria, but biofilms are also prominent in causing gum disease, periodontitis, also an oral disease, but different from cavities or caries. Okay, so cell walls, these are tough, these are protective, these protect the inside from the outside. Keep in mind that the cell envelope is the cell wall plus the cell membrane. The cell membrane is a bilayer of phospholipids. The cell wall is outside and differs from different types of bacteria. We talked about gram-positives, gram-negatives, mycobacteria a little bit. All these have different cell walls, but the same plasma membrane. So the book talks a little bit about some osmotic pressure. So the cell wall is going to help keep the cell from becoming too full of water and blowing up through a process called lysis. So the cell wall is relatively porous to substances coming across, whereas the cell membrane is less porous and is going to be more selective about what's going to go across it. So this can happen by two means. Either, let's see, that's coming up in a second, but I'll go ahead and mention it while I'm on it instead of dropping the thought. So this can happen through facilitated diffusion, or even just diffusion or active transport. Diffusion is moving from high to low concentration. Active transport is moving from low to high concentration. Did I say that right? High to low, diffusion, low to high, active, against a gradient. Okay, let's talk about gram-positives, gram-negatives. We mentioned these a handful of times. Now we're really going to get into what is a gram-positive, what is a gram-negative. It's based on the cell wall structure, okay? That's how we're going to organize these. So gram-positives have a really thick outer layer called peptidoglycan in their cell wall. It also has something called teichoic acid. We don't really understand the function of teichoic acid, but if you notice in this image here, it is going to run at a right angle to the peptidoglycan, so it might help strengthen the peptidoglycan layer. So provide some sort of rigid structure to the cell wall. Peptidoglycan is a very rigid outer structure though, and in gram-positives, we have a very thick layer. Gram-negatives also have peptidoglycan, but it's very, very thin. The book describes it as a two-dimensional layer, meaning it is just a single, like a pleat, a single layer, as opposed to a thick layer like gram-positives. I'd prefer to just say it is thinner versus thicker. And gram-negatives do not have teichoic acid. Again, if teichoic acid plays a role in maintaining structure of peptidoglycan, you don't have as much of that, you don't need teichoic acid. This shows our gram-negative cell wall. So notice here we have this really thin peptidoglycan layer outside of our plasma membrane, but then we have this outer membrane that is not present in gram-positives, and the major component of this is lipopolysaccharide. So lipo refers to a lipid, so we have lipid A, and then polysaccharide, it is a large chain of sugars. there's also a lipid component to it. So LPS lipopolysaccharide, only gram negatives, never found in gram positives. And since we have two membranes now, this inner and outer membrane, in between them, where the peptidoglycan is, we have what's called the periplasmic space. So periplasm, between the two membranes. And then this outer membrane with the LPS contains some porin to selectively allow some things into the periplasm, which can allow things to be actively or diffusely transported across the plasma membrane. But we have two membranes now instead of one. But again, the distinguishing factor is that gram negatives have LPS. I pulled this image from a paper just because we've mentioned mycobacteria a handful of times. So I want to show you why I would argue mycobacteria do not fit within a gram category. The book goes back and forth in this chapter, and it was kind of annoying, honestly, about how mycobacteria fit within, I believe it was the firmicutes. So it says that they belong to a gram-positive category, but then it later says that they are more closely related to gram-negatives than they are to gram-positives. But they don't follow a gram staining technique. So taxonomically, they might be considered closer to gram-positives, but practically speaking, for our purposes, let's say that they are distinct. They are their own type. They are acid-fast positive. They are not gram-positive or gram-negative. They are acid-fast positive. So what an acid-fast test looks for is this outer waxy layer called mycolic acid. This is not present in gram-positive or gram-negatives. This is only in mycobacteria, only acid-fast organisms. Okay, so just like in our bacteria, the archaeal cell wall is used for mechanical strength, so for protection from the external environment, but it lacks peptidoglycan. It has something that resembles it, but it's chemically different, called pseudo-peptidoglycan. It's kind of like peptidoglycan. I do not need you to know chemically how it's different, just know that it's not the same. This isn't a chemistry class, so I don't need you to memorize the chemical differences. And keep in mind that we can also have some sugars or proteins in the cell wall also. They will also have a surface layer or an S layer with proteins and glycoproteins, so sugars attached to proteins. And this is going to be in a really organized lattice. Some bacteria also have an S layer. Not the most important thing we're going to talk about today, though. So again, this helps keep their cellular integrity and protect them from their external environment. Okay, so we mentioned that certain molecules can diffuse across a plasma membrane, others have to be actively transported across it, and that can take place through different types of membrane proteins. Within a cell membrane, I would like for you all to view this as a very fluid structure. Don't think of this as a rigid, fixed. The proteins are moving. Think of it as an ocean, as a sea. Things are always moving. It's very fluid. It is not fixed and stuck in place. And so we call this the fluid mosaic model. Mosaic because from one second to another, if you were to take a snapshot, the membrane could look different because it is very fluid and moving. So we've mentioned that the phospholipid bilayer has hydrophilic heads on the outside of the cell and on the inside, and the bilayer has hydrophobic tails on the insides of the bilayer. So this makes it hydrophobic in the middle of the plasma membrane. So if molecules are trying to cross, they have to go through a hydrophobic region. So we can use something like a integral membrane protein that is going to span the bilayer, and that can allow a channel for molecules to cross, so they don't have to go through a hydrophobic region. So the difference between integral membrane proteins and a peripheral membrane protein is that integral membrane proteins span the bilayer. They're going to go across both layers. Whereas peripheral membrane proteins will not go through both layers. They'll be attached to the head group, or I believe the book says they might even be attached to the outside of an integral membrane protein, but they're not going to span the bilayer. They don't go into the cell. They're only on the outside. So some antimicrobials are going to work to either poke holes or even disrupt entirely the plasma membrane. So polymyxin is going to poke holes, whereas something like ethanol will just dissolve a plasma membrane. So we have some proteins in the membrane that can help synthesize the cell wall. Keep in mind that enzymes are proteins. They can produce energy, help with DNA replication, or recognition of external stimuli. All right, and then when we're transporting across a plasma membrane, I've mentioned this once, but while we have it on the slide, I'll do it again. So we can either do it passively through facilitated diffusion, so going from high to low concentration, or going against a gradient from low to high by using energy to move molecules across the membrane. And that's called active transport. Okay. This is a small little box. We're just about done. The archaeal cells have some differences from bacterial and eukaryotic cells. Notice here that the linkage to glycerol is a little bit different. We have an ether instead of an ester. I don't need you to memorize that. Just know that it's built differently, conceptually speaking. There are usually no fatty acids. Instead, we're going to have these 5-carbon repeats. And we can even end up with a bound monolayer as opposed to a bilayer in some archaeal cells. But that is not going to be present in bacterial or eukaryotic cells. So what I want you to know here is just that there are some differences between bacterial and archaeal cell walls. I'd say the biggest difference is that some archaea can have a monolayer, whereas you're not going to see that in bacteria. That is the major distinction I want you to get from this slide. Yeah, so the cell membrane of some archaeal species can be a monolayer, so a single layer, as opposed to a bilayer of phospholipids that is going to be present in all bacterial species, as well as eukaryotes. So that is special, distinct among Archaea. And again, if you're looking for this in the book, it came from this Microfocus 4.3 small box. All right, we are getting close to time. We have about 10 minutes before the quiz, so let's try and wrap this up. Let's see where we're at. All right, we're getting there. Keep in mind, bacterial cells have a nucleoid where the DNA is located. They lack a membrane-bound nucleus, like eukaryotes have. They are using DNA in all cases for their genetic material. And the complete set of genes within the DNA in any organism is called a genome. Not a chromosome, because humans have more than one chromosome. Bacteria can have a plasmid. We need to include that as well. So all of the genes in an organism is called the genome. If you're talking about all the proteins, it would be the proteome. All of the transcript is the transcriptome. You're just going to add "ome" to the end of something, and that refers to all of it. Call it the age of omics. So if you only have one copy of your DNA, you are haploid. Humans have 2 copies of every chromosome, so we are diploid. Some bacteria have plasmids. These are really small, round bits of DNA. They might have anywhere from 5 to 10 genes on them. So these can be used either intentionally in a lab, or bacteria can transfer them through those conjugation pili. They can inject a plasmid into another organism, and that can do something like allow it to have genes for antibiotic resistance. So if you need this organism to stay alive, you can give it genes to help you stay alive as well. It'd be the equivalent of if I was really hungry and couldn't teach and you all brought me some food, for example. It's not the exact same, but it's kind of like that. Okay, all proteins are going to be synthesized, eukaryotes, prokaryotes, through ribosomes. They're smaller in bacteria. And if you are in the cytoplasm, you're going to make proteins for the cytoplasm. Others are going to be associated with the membrane, and they're going to make proteins for the membrane or to secrete out of a cell. 2 antibiotics that are going to inhibit ribosome function are streptomycin and tetracycline. You'll have probably heard of both of these. If you don't have ribosomes, you can't make proteins. You can't make proteins, you're dead. Just to briefly mention these, we have some microcompartments. So these are wrapped in proteins. These are unique to bacteria, so Archaea don't have these. Some cyanobacteria have something called a carboxysome, which is where photosynthesis will take place. So CO2 can be converted to sugars in these carboxysomes. They can also limit the diffusion of volatile or nasty substances. So basically, if you have a nice weapon inside of your cell, you can keep it coated in protein so that you don't end up hurting yourself. Think of this as having a lighter in your pocket. You don't want it to be on fire in your pocket, because then you would feel that burn too. We can also have inclusion bodies to hold things like glycogen, sulfur, phosphate, calcium. We mentioned this on that large blown up slide that we viewed earlier with the pili and the flagella. So some aquatic organisms are going to use gas vesicles to adjust their buoyancy. So if you have gas in a bubble, you can float up the water column. If you get rid of that gas bubble, you'll float down in the water column, just like scuba diving. And then some organisms use magnetosomes to detect magnetic fields so that they can get away from oxygen. We need oxygen. Some organisms die in the face of oxygen. So all cells, bacterial and archaeal, are going to have a cytoskeleton just like us. It's a little bit different. What I want you to know is that within the cytoskeleton, we can have some parts that are used for cell division, others are for chromosome segregation, whereas others are just to maintain the structural integrity of a cell. But all cells will have a cytoskeleton present. Cytoplasm, then you're going to make proteins for the cytoplasm. If you're a ribosome on the cell membrane, you're going to make proteins for the cell membrane, cell wall, or for secretion. So that is what we have. We are right on time, everybody. So we're going to go ahead and take our quiz. So I'm going to ask that we all stay logged in, and if you have your webcam off, please turn it on. For the quiz, it's going to become available in 4 minutes, so at 11. So you'll have from 11 to 11:30. I'm going to watch everybody's webcam. Basically, please don't have your book open. Of course, I can't come and knock it out of your hands, but we're here to learn, not to get good grades. I understand you want good grades, but please be academically honest. With you all being at home, there's only so much I can do to monitor you. So if during the quiz you have any questions, I would request that you type it in a little chat box so that we don't have to interrupt others. But I'll mention it again. I did take out the 2 hardest questions, so it is only 10. Really hard this weekend because I've intentionally made this not very hard. If you struggle with this, reach out to me afterwards and we'll figure out a way to get you back on track, okay? So I'm gonna go ahead and stop talking so that you all can open this up, and I'm gonna be available for the next half hour while you take your quiz. I will check my email if you have any problems there too, okay? Questions? Anybody?
Can we just log off after we're done with our quiz?
Yes. Once you're done with your quiz, go ahead and log out. And if you have any problems you want to address, just shoot me an email.
Okay. For some reason, my like video option is not popping up. So can I like try rejoining the meeting to see if it'll pop up?
Yeah, do that, because I'm not going anywhere, and 30 minutes is more than enough time to answer these 10 questions.
Okay. Thank you.
Where are we finding the quiz?
I'm sorry, what's that?
Where are we finding the quiz?
Oh, the quiz. So the quiz is in Canvas on the left. So let's see. In our class on Canvas, on the left, you should see quizzes, and then it says quiz 1. Can you see that?
Yes.
Okay, great. So it should be live in 1 minute, and there's no password required for it or anything like that. You are going to get 1 chance to finish this, and I have it set up so that you'll get your answers. You'll get feedback immediately, and then you'll get the correct answers available. Oh, sorry. Wait, did you say that I'm muted? No?
No, I can hear you.
Okay, great. That's what I thought. So you are going to get your responses back as soon as you submit it, and then the answers are going to be available in a half hour afterwards at noon today. So you can log back in and see what the correct answers are and use that to study. All right. So it looks like we are just about at 11. So good luck, everybody. Let me know if you have any issues as you work on it, okay? You can do it if you want, or you can go out there. You can do whatever you want. Yeah, you might as well. I'm here. He's here. I'm going to run to the restroom. So everybody getting without a problem?
You said answers would be available at the end.
Mm-hmm. Feel like you did okay?